I'm very happy to announce our last speed round of the day and introduce our session chair for this session, who you've met earlier, Dr. Patrick Viethuybeck, who's the Deputy Radiologist and Chief and Director of the PET-MR program at the Joint Department of Medical Imaging. Please welcome Patrick.
Home stretch, the last two sessions. And first one would be, as Melody said, our speed rounds combining radiotheranostics with research innovation of tomorrow. We have several distinguished speakers. And the first one would be Dr. Karen Stephenson. She's the Director of the Nuclear Research and Education Support for Nuclear Operational Facilities at McMaster. She holds a PhD from McMaster in chemistry and completed her postdoctoral fellowships in radiology at UPenn and PET Center of Addiction and Mental Health here in Toronto. More of 20 years of experience in this industry in nuclear research. And before joining McMaster, she was at CPDC, led there the research and discovery of new radiochemistry and radiopharmaceuticals. And she is currently also the CSO of the Canadian Nuclear Isotope Council and a board member of Nuclear Innovation Institute and the Canadian Medical Isotope Ecosystem, one of our partners here. I would like to welcome Dr. Stephenson to the stage. Thank you.
So I want to thank the organizers for inviting me today. I'm really happy to be here to talk about something that I'm really passionate about, and it's a little bit of an overview of how research is underpinning some of the innovation and commercialization that's happened in Canada. I think we can all agree that the commercialization pathway is fraught with challenges. When we look at discovery science and isotope science, it really the basis of having isotopes is critical along the whole pathway. And we want to make sure that we're lucky here in Canada that we have access to all of the isotopes. We have a fleet of cyclotrons. We have the research reactor at McMaster, as well as now we have our CANDU power plants also able to make isotopes. So it's important that we have that baseline of research capability in order to move through the pathway. If we move along, it's really through preclinical research and discovery science that is iterative until we move forward with the next stage, which is the clinical research will validate, and we've talked a lot about this today, but it really is what leads to the commercialization is through clinical research activity. We have a lot of discovery science that will go back into a true candidate because we have to imagine how we're going to scale that up to provide it reliably and provide access and make commercialization feasible. And finally, there has to be sustainable innovation and product lifecycle management. But mostly what I'm excited to talk about is the radioisotope R&D history in Canada. And I'm sure some of you may know some of this, and I might be missing a few, but hopefully I'll capture all of the highlights. If we go back as far as 1940 and looking at the production of radioisotopes in Canada, that's almost when Marie Curie was discovering them. So we're pretty much at the cutting edge of isotope development and understanding how to do that in Canada. We had the NRX reactor, the NRU reactor, and the MNR reactor come online during that time period, as well as a number of different isotopes. In particular, we are also, in 1951, the first cobalt-60 medical treatment happened. They called it the cobalt bomb, which is pretty clever. It's still used today. It's pretty great. If we look at the next 2 decades, the NRX started producing I-125. We brought Molly online. And interestingly, most of you would think F-18's made in a cyclotron, but this actually was made in the reactor. And it was on the cover of Nature magazine in the production of the radiofluorination of F-DOPA. And it's actually said that that early work in dopamine really shaped the broader fields of radiochemistry, molecular imaging, and drug discovery, as it was really fundamental science at that time. Between 1980 and 2000, we had the production of Y90 TheraSphere come to fruition. It's the most studied evidence-based Y90 therapy to date. When we get to 2000 and 2020, we're starting to get into this radiotheranostics renaissance. Unfortunately, this is when the NRU is shut down, but there's always opportunity when things happen like this, and that brought the CANDU power reactors and looking at that. And most strikingly, I always lead to this image that was Image of the Year in 2018, and for the first time, it was really striking to see the impact of lutetium-177 PSMA 617. It's impressive, and it still is something we're all talking about today. Iodine-125 production at McMaster, so this was research that was happening in the 90s. The patent was filed in 1997. It kept the lights on in the reactor and allowed us to still be able to then expand and do more, make more radioisotopes that we could never have made if the reactor had been shut down. We have scaled that process up, and now over 1 million patients have been treated with I-125 at McMaster. But I think what it also did is it kept our nuclear facilities functioning and growing. And that led us to enabling a pipeline of spin-outs that we all talk about today and some of the greatest success stories that have come out of Canada. We were home to the Center for Probe Development Commercialization, which was started by McMaster faculty member John Valiant. Adam V is also hosted at McMaster, as well as Fusion Pharmaceuticals. Both are spin-outs of the CPDC. So lots of success has been tied to the fact that we kept those facilities going at McMaster. I'm going to go across Canada and give you some examples. I think if we look to the West Coast, Artemis is one of the best examples. It was in response to the technetium crisis in 2009-2011 that led to the foundational research and the starting of the company by Dr. Paul Schaefer and scientists at Triumph. They have expanded that production between 2016 and 2024, have solid targets for isotopes that we all like to have access to today, gallium-68, zirconium-89, copper-64. They were acquired in 2024 by Telix Pharmaceuticals for $82 million US. That's an impressive translational approach from basic science and research. I think probably one of the biggest impacts globally will be the isotope production system that's gone into Bruce Power. The construction happened in 2002. They first came online and were producing lutetium-177. They've now had a robust, reliable source of lutetium-177 that provides that security for pharmaceutical companies to invest and make sure that patients will always have access. In addition to that, it also provided an opportunity for historic partnerships with the Saugeen Ojibwe Nation for them to jointly market isotopes in support of the global fight against cancer, but to also create new economic opportunities for the San. So it has a huge impact in many different ways. Fusion Pharmaceuticals, I've talked a little bit about, but in their technology, they are a next-generation precision cancer medicine company. The main technology that led to their success was the development of the fast clear linker. And I've shown the image here that you can see one of the most important things is that the clearance rate of their linker system is much faster than commercial options. What that means is that you can remove all of the background target faster, which is a really important aspect to development of a theranostic. It is a spin-out of CPDC and supported at McMaster University and was acquired in 2024 by AstraZeneca, as I think most of us know, for, I think, a finalized value of over $2 billion. If we go out to the East Coast now, we can see a really great example out of Dalhousie University. The fundamental research was developed there and patented. They developed these glass microspheres that you can now visualize through X-ray and CT, and they're radio-labeled with Y90. They had their first patients have been successfully treated in 2024, and it will be really exciting to see where this particular company goes in this technology. So I've highlighted kind of decades by decades, but I want to just point out what has happened over the last 5 years in Canada. It's really impressive in terms of radioisotope development and research. We have Bruce Power, I already mentioned. We had the first customer delivery of actinium-225 by Actinier. We had the historic BWXT, Laurentis Energy, and OPG production of Moly-99. That's bringing the production of technetium-99M back into Canada. Kinetrix moved down the value chain in a positive way. They're now able to produce the enriched target material. It's really hard to get an enriched target for those of us that are making radioisotopes. It's very expensive and challenging to get, so it's wonderful that that's being developed in Canada. As well as through this time, it was also COVID. And what we discovered is, most of you will know, our radioisotopes are shipped on passenger planes around the world. I know for us in shipping I-125, it was a real challenge. We were approached by Air Canada Cargo and said, I think we can help you. Let's try to use drones. I didn't think that was going to work. And we've actually successfully now shipped lutetium-177 from Milton to the Oakville Trafalgar Hospital and been able to demonstrate that at some point when we need to execute this technology, we'll be able to do so. So I always want to look forward and what is the next emerging innovation. I think there's a couple of areas that are really exciting. In this room, in my role, I look a lot at the energy side as well as radioisotope production, and it's really interesting what's happening with small modular reactors. We have a neutron shortage in the world. They're going to be producing neutrons as well. Can we use them to also make isotopes? They're thinking about it as well. This is a company out in New Brunswick called Arc Clean Innovation Technology, and they're looking at that. And I also want to point out a technology I'm really excited about is Astrosystem. It's a fusion-fission based production. I know they're here today. It's really exciting to imagine where fusion-based neutrons will go, small modular reactors neutrons will go. I think we're going to have a lot of different ways to make radioisotopes in the future. So I'm a chemist. I have to show the periodic table. It's exciting. So if you can look here, so those that are in green are those that are sort of routinely available. Those that are in red are those that are exciting or emerging, and maybe they will take off. But it's exciting to imagine what the future might be. Will you have a menu of isotopes that you can pick from and then use it for the application to really get to a precision medicine approach? And so I think the future is exciting for Canada. We have lots of great stuff happening. Thank you so much.
So maybe we have time for one question, if there's one question from the audience. Maybe. No, please. Or you just speak very loudly.
Sure. So I'm obviously the CBDC fusion AbbVie success change is such a great story that comes out of McMaster. From the perspective coming from an academic institution, looking at all the great academic institutions that Canada has, what can they do to potentially encourage or foster from an institutional level perspective more commercial activity and more potential startups and spin-outs coming out of the academic?
Sure. So I think there's a couple of things that we do at McMaster that might be translatable. One is that is our licensing framework. So when a company or startup company is looking to launch and get going or translate their activity, they often don't understand the Canadian licensing framework, and they often struggle with the health physics and the safety perspective. So one of the things we do is we lower that barrier of entry. We do all of that so that they just have to worry about their science and how to translate it and scale it. So making that accessible is a big win. I think the other part is just encouraging to the viewpoint of not being held up by the patenting process, but really to try to imagine in events like this and collisions and letting people see and network and talk. I think people need to be excited a little bit to take the leap of faith. So I think Professor John Valiant, he was my supervisor. He was a bit of a unicorn in some ways too. He was really brave, I'll say. Yeah.
Thank you. All right, next speaker is Dr. Meena Swiha. He's a dual-trained physician in oncology and nuclear medicine, which is actually very cool. And he did residency training in Egypt for clinical oncology, and afterwards did a PhD focused on breast cancer. Then further on went to residency in nuclear medicine at the University of Western Ontario, and afterwards did another clinical fellowship for prostate theranostics in St. Vincent's Hospital in Sydney in Australia. Very nice place. Primary research currently interest focuses on, obviously, prostate theranostics, FAP imaging and FAP theranostics, as well as neuroendocrine tumors and thyroid cancer. Dr. Swiha. Coming from very far away.
Thanks, Patrick, thanks so much for the invitation and for the introduction. So today I'm going to talk about lutetium PSMA 617 in combination and adaptive dosing. Very interesting topic. Lutetium PSMA 177 is a small molecule which is used for treatment of prostate cancer. It emits high energy beta radiation to prostate cancer cells expressing PSMA receptor with activity and safety. Vision trial is one of the most important clinical trials. It's an international phase 3 randomized trial, included more than 800 patients with late metastatic castrate-resistant prostate cancer, both ARBI and chemotherapy, and it showed significant improvement of the overall survival by around 4 months in patients treated with lutetium PSMA compared to protocol-permitted standard of care. Moreover, the therapy trial, it's a multi-center phase 2 randomized trial, include around 200 patients in Australia, and the trial compared lutetium PSMA 617 to a strong comparator cabazitaxel in late metastatic castrate-resistant prostate cancer, and it shows that lutetium PSMA 617 improved the PSA response to 66% versus 37%. It also improves the progression-free survival and the quality of life. Many of us here in the room might recognize this image. This is the SNMI 2018 image of the year presented by Professor Michael Hoffman. And the image shows 8 patients with mCRPC treated with lutetium PSMA, and all the patients achieved more than 90% response in PSA, in addition to significant radiographic response using PSMA PET. Despite all of this, we have approximately 1/3 of our patients who will have upfront resistance to lutetium, so lutetium 177 PSMA 617, or they will have a limited duration of response as per both vision and therapy trial. So this is one of the early patients in Ontario who was treated with publicly funded Pluvicto, and the patient is 73 years old gentleman. As you can see, he has mCRPC to extensive lymph nodes above below diaphragm, bone, and liver. He has progressive disease after abiraterone, acetate, and docetaxel. Careful examination of the baseline PSMA PET and contrast-enhanced CT showed no PSMA negative measurable disease. The SUV max of the most avid lesion was around 55, which is very high, and the disease looks visually homogeneous. And in LHC, we use visual scoring to predict the SUV mean, and thus we can predict also the response to lutetium PSMA, and we use the heterogeneity and intensity of tumor scoring, HIT score, and the patient is HIT score 3, which means he has the second highest quartile of SUV mean between 8.5 to 10. So the patient started treatment. He received 3 doses of lutetium PSMA. PSA responded nicely. As we can see, it dropped by around 43%. And after the 3rd dose, we repeated, as we decided for all the patients, we will do radiographic assessment of response using PSMA PET-CT in addition to contrast-enhanced. First look to the scan can show to you, okay, there is good response to the disease. But a careful examination to the scan, it will show mixed response. The patient has significant response in the PSMA-avid disease, in the nodal lesions and some other stuff, okay, and he developed new low PSMA avidity disease extensive within the bone marrow. You can see right now the extensive involvement of the bone marrow, in addition to new liver lesions confirmed by contrast-enhanced CT. Repeat PSA after the PSMA PET showed rising of PSA to 36. Can we do any better? Prostate cancer is known for its heterogeneous nature. It has significant heterogeneity, both intertumoral and intratumoral, as been many clinical trials in genomic and phenotypic heterogeneity, and this is due to accumulation of multiple mutations. This data drove multiple clinical trials addressing the combination of lutetium PSMA with other effective therapies such as immunotherapy, DNA damage repair inhibitors, chemotherapy, and ARBI. Single-agent immunotherapy has limited efficacy in the mCRPC conditions, in addition to its significant toxicity, although there is growing evidence that radiation can enhance the impact or the efficacy of immunotherapy by modulating the tumor microenvironment. Few trials or several trials addressed this, such as PRINCE trial in combination with pembrolizumab and lutetium PSMA, and also Evolution trial, which has combination between lutetium PSMA and Ipi/Nivo. Both trials showed significant toxicity. So 14% of the patients in the PRINCE trial has to stop pembrolizumab, and the Evolution study has to stop recruitment earlier due to development of serious side effects. PARP inhibitor is very attractive also. So PARP inhibitors show by the profound trial, it showed efficacy in MRCP, metastatic castrate-resistant prostate cancer. It has the ability to convert single-strand DNA breaks into double-strand DNA breaks, but in patients who has BRCA1, BRCA2, or ATM mutations. So what about with radiation? So the LUPARP study, it's a phase 1, phase 2 trial in Australia, which has combination olaparib and lutetium PSMA address this. We know that beta particles like lutetium primary cause single strand, so the combination even might be more effective. We are still waiting for the results. Very effective combination is a combination between lutetium PSMA and chemotherapy. Concurrent chemoradiotherapy has been the standard of care for many tumors, such as esophageal tumor and cancer rectum and lung cancer. So what about here? In the VISION trial, although lutetium was superior to cabazitaxel in both radiographic progression-free survival, PSA response rate, and quality of life, but there was no significant difference in the overall survival. This means that both treatments are effective. So what about the combination? So we know that chemotherapy can act as a radiosensitizer in addition to its own cytotoxic effect. And this was the rationale for the LUCAP study. It's a phase 1, phase 2 trial, dose escalation, dose expansion in Peter McMullen in Melbourne. And they used the combination between lutetium PSMA 617 and cabazitaxel, still waiting. However, when we discuss combination between lutetium PSMA or radiation and chemotherapy, we have to think, hypothetically, the combination will have more efficacy, or we will have like higher response rate. But the most important thing, what about the longevity of the overall survival and the quality of life in those patients? Which one is better? Having the combination or sequencing? Like, for example, the new trial, one of the trials which was recently published is the upfront PSMA, but this one was in the metastatic hormone-sensitive prostate cancer using lutetium followed by the docetaxel. One of the most attractive and approved most attractive trials, addressing the combination with lutetium PSMA is the ENZABI trial. ENZABI trial, Australian trial, multi-center, phase 2 randomized, include around more than 160 patients with early metastatic castrate-resistant prostate cancer prior to chemotherapy. The patient had 2 or more high-risk factors for having failure on enzalutamide. And the patient were randomized either to enzalutamide full dose 160 milligram versus enzalutamide plus lutetium PSMA 2 to 4 doses. And early work have shown that patients with metastatic castrate-resistant prostate cancer treated with novel hormonal therapy or ARBI, many patients or some patients might have upregulation of the PSMA receptor. And this upregulation can be easily explained. We are treating heterogeneous group of cells. So there are hormonal sensitive cells like the blue ones, and there are resistant cells like the yellow ones. If we treated the patient with novel hormonal agents like ARBI only, so what's going to happen? We are going to control only the hormonal sensitive. However, on the other hand, nasty cells which have alternative growth pathways, such as the PI3K act immature, which is associated with PSMA overexpression, they will proliferate. Can we mitigate this by adding lutetium PSMA? And adding lutetium PSMA can control diverse clonal population, as we can see, in addition to the upregulation itself, which might add to delivery of higher doses of radiation to the cancer cells. The ENZABI trial showed significant efficacy. If we can see here, the PSA 50% response rate reached 93%. If you go back to the therapy trial, the response rate was 66%. This is significant improvement. The PSA 90% response was almost 78%. ENZABI trial showed significant improvement of both radiographic, the radiographic and PSA progression-free survival in addition to the overall survival. The benefit in the overall survival reached 8 months, which is almost double what we got from the VISION trial, around 4 months. I think one of the most important things in the therapy trial, in addition to being like the first randomized phase 2 trial in combination, the remainder of the combination trials, most of them were phase 1, phase 2, but this one is a randomized phase 2, is this is the first structured trial which has early response assessment and adaptive dosing. So it's a huge database. The patient had multi-time point imaging with PSMA PET scan, baseline day 15 to prove the PSMA upregulation, and they have SPECT-CT 24-hour after each dose of lutetium, in addition to day 92 or week 12 between cycle 2 and cycle 3, they have PSMA PET scan for response assessment. So at week 12 before the third dose, patients who still have persistent PSMA positive disease on the PET scan with uptake above the blood pool activity, they were offered 2 additional doses of lutetium PSMA to a total 4 cycles. And patients with no persistent disease above the blood pool activity, they discontinued lutetium at 2 cycles. This is the first trial addressing the adaptive dosing and early response assessment, although there has been some early work in the therapy trial. 7% of the patients were offered treatment break after having significant response. And there was like a retrospective analysis called RESPECT study done by the same team in Sydney, which offered the patient breaks, and the patient achieved after like second SPECT to have like significant response on both PSMA and SPECT-CT. They have longer survival up to 19 months, which is theoretically above what we got from the VISION trial, but this is a retrospective analysis, so we cannot count on it. To conclude, combining lutetium PSMA with other therapeutic agents may enhance the response by targeting diverse clonal population and acting as radiosensitizer. Early response assessment using molecular imaging by PET or SPECT, in addition to the conventional imaging, enables early detection of treatment resistance and treatment adaptation. Personalized and adaptive dosing using early response biomarker has the potential to achieve similar treatment response to the standardized dosing. Thank you so much.
Thank you. We have again time for one question. Otherwise, there's some time after all the other talks for some questions and answers as well. I certainly would have one question. Do you think, there's always the talk about sequencing versus combination of therapy. Currently, we're using it as single modality therapy, at least in prostate cancer. But do you think that eventually all theranostics, at least prostate theranostics, whether it's with actinium or lutetium, all end up in combination of therapy, or would you see that there is one or the other indication left where we just treat with radiopharmaceuticals?
Excellent question. Thank you so much. So I think combination therapy will be an important role in the coming era, because the problem is that we have still significant failure rate on it, especially with the combinations which are safe, like, for example, with hormonal agents and PARP inhibitors. I would expect these ones will grow. With chemotherapy, still we have to have enough data about sequencing, okay, and overall survival. It will be a little bit early because of the significant toxicity you might get from it. But I think we are moving towards this direction.
Okay. Thank you very much.
Thanks. Thanks.
Our next speaker is Dr. Ilya Gibb. From industry this time. He's the Oncology Chief Medical Officer at GE Healthcare, and he is helping shaping the strategy of this entity, since it's like a spin-off from the overall GE conglomerate, and helping to connect customers, obviously, and shaping the strategy with healthcare systems, tailoring solutions for cancer care. He also collaborates very closely with the leaders in the healthcare system and governmental bodies worldwide in diverse projects aimed obviously in advancing medicine, especially in cancer care. Dr. Gibb also contributes his expertise as a board member to Trova Now, which is a non-profit organization or consortium dedicated to pancreatic cancer early detection and treatment, and another non-profit organization, which is the Florida Lung Health Coalition, also for improving population outcomes for lung cancer. Please welcome Dr. Gibb.
I'm so honored to stand before you and talk about data-driven radioligand therapy and strengthening the role in cancer treatment landscape. Even though I am employed by the healthcare industry, I am still a practicing physician, and this talk is not intended to be commercial, and I hope it's going to go as intended. First of all, I'm going to state something quite obvious. Radiotheranostics is coming into the space that is not a vacuum. There's already other treatments that are available, and I think a lot's been already said today about the combination potential and synergizing the role of radiotherapy and radioligand therapy with other therapies as is. So radiotheranostics is on the far right. Obviously, this is not an extensive list, but we can expect a lot more to come, and I think that this also by itself implies a certain challenges to what I'm going to be talking about and the necessity for the data to be present. Now, I think it already became a tradition, so in that this picture has to be shown, and so I'm following this tradition. But the thing what I'm going to talk about in this slide is actually not the fact that there are super responders, because I think for almost any therapy there are super responders, but we don't want this to be a lottery win. Therefore, I looked at some of the papers, and this paper came out just a few months ago, talking about that up to 50% of patients who receive lutetium PSMA do not experience a PSA response. And I think that this is something that we need to focus, not just because of the clinical aspect and the necessity to solve this in a way and form that we are much more predictive and clinical efficacy is not an obstacle, but actually an advance to the treatment, but also in the landscape of the payment models that I think are inevitably coming into our space. And I think you might be familiar. If you're not, then I hope this also going to be useful, is the term OBA, which is outcome-based agreements. Once getting ready for this presentation, I looked at the availability of Canadian data, and I was actually very pleasantly surprised because in 2021, there was the paper talking about outcome-based agreements, an overview, and one Canadian province, Alberta, was among the top countries that adopted this innovation. So what outcomes-based agreements are is instead of being paid per treatment, the payment and reimbursement is coming per patient outcomes. And I think that this is, for now, not necessarily connected to radioligand therapies, but considering the expense of radioligand therapy, there is already multiple entities, payers, countries that are adopting and integrating those payment systems for radioligand therapies. What I did then is I looked at the same authors and I followed them, and sure enough, a couple of years after, they looked and they published another paper that was talking on what are the metrics that they used and that potentially can be used specifically in Canada and actually linked to oncology space. So I don't know if you can see it, it's a little bit small, but number 1, number 2, and number 4, so, number 1, number 3 is overall survival, progression-free survival are already the adopted ones, and number 4 is patient-reported outcomes that at the moment of the publication was a no, and it's not a part of the metrics that is defining how systems are being reimbursed for the cost of the therapy. However, it has a potential, and it should be in the future, which also is, in my view, important and interesting enough. Now then going next, there was another publication that came out just last year, also in the Canadian space, and talking about financial characteristics, what the Canadian payers and pharmaceutical manufacturers prefer. That's an interesting review because it's not exactly linked to oncology space, and by far both of these slides are not exactly linked to radioligand therapies. Nevertheless, it's talking about various metrics and the various model names that have an effect on the payment structure for radioligand therapies. Again, it's not just about the clinical space, it is also about the availability of the treatments for the patients. Because if we're spending those drugs and money on patients with potentially non-responders, I think it becomes not available for patients that could be a better candidates. So it's also about the efficiency and the value-based care, which is somewhere within those models. So you can see that on the short term, there is a discontinuation. So if patient provided is discontinued the treatment by a specific milestone, there is non-responder rebates, and there is also free trial periods on the short term, but there is also a medium long-term duration of those models that have an effect, and in my view, correlates very well with radioligand therapy. Now, the reason I spend so much on talking about the payment is because I think this also is going to have an effect on where radioligand therapy develops in the future. So in order to answer that, and in order to start looking at how and what can be done in terms of having better outcomes for radioligand therapy, number 1 is individualization. Not all patients are the same. Then why is the dose the same, and why is the amount of activity that is given the patient is the same? I think this is quite obvious. Nevertheless, every time I bring this or something similar to this into the audiences like that, I always get some positive feedback. It's like, this needs to be done. Okay, so individualization is part number 1. The other thing that needs to happen, And obviously, this is also data-based. The other part is personalization. And for this, I just looked at a couple of papers, looking at different patients, looking at the potential dosimetry and the volumetrics that is done to the patients. And you can see that there is different volume, there is a different way of absorption, there is different way of being depicting on the changes in the body, as well as there's different profiles of those patients. The lesions can be homogeneous, they can be heterogeneous, and I think this all is having a certain effect. Now, if there is so many different dependencies, I talked about individualization, I talked about personalization, okay, well, with all these dependencies, if I want to be less dependent on the weather, I think the answer is there's got to be a great weather forecast. It's got to be a great prediction in what the weather is going to be like, making me less dependent and less affected by the weather, what I dislike. But there is one other component to this. It's not just that the weather forecast got to be great for tomorrow. If something happens tomorrow, I got to be also receiving a better weather forecast for a couple of days down the line. So therefore, looking, for example, at this dependency where lesion absorbed dose decreases in the next cycles. So in the graph on the right, you see the blue bar, which is the first cycle, and then it's decreasing for various lesions in cycle number 2, cycle number 3. So that's another dependency. And I can keep going. It's just that I'm limited by the time and the clock ticking, that there is a lot more of those dependencies. Now, in order to be able to also look, and this is making it a little bit easier for me because I saw the slide, something like the blueprint and expanding the blueprint earlier with one of the speakers today. I think it's going to basically just repeat this. So whether it's alpha particles, beta particles, Augers, okay, well, this is one combination. The other one is whether it's small molecules or maybe larger molecules or antibodies is going to come into the future. There is multiple radioconjugates that, when you combine one with another, is already making this multiplied by itself. And then there is multiple sequences, whether that's a single treatment, multiple treatment, how is this going to be in the world of adaptive radioligand therapies, and multiple synergies. Okay, we already talked about the fact that it's not coming into the vacuum. And if I multiply all of these together, I'm coming up to thousands of different combinations. So the complexity is there, and evidence is needed to be driving those adoptions and to make sure that those clinical efficacies, financial efficacies, are not affected by all this complexity. So coming in next in what can be done and what do we believe is valuable to do. Number one, there is various consortiums, and this is an example actually coming from Europe. It's set by the Innovative Health Initiative in European Union, and it's partially using European Union funds. But the whole idea for this Thera4Care consortium is driving diagnostic innovation, therapeutic precision, sustainable supply chain, which a lot has been said already today, sharing the knowledge and training, and among other is data-driven insights, not just by bringing in all this data, but making sure that this data is actionable and can be used for what I just described in the previous few slides. The other part is promoting the dialogue. Just about a month ago, I had the privilege of arranging another non-commercial discussion synergizing radiotherapy and radioligand therapy. Yes, there were representatives from the industry like Elekta, like MIM Software, but there were clinicians, there were IAEA. And it's interesting because in general, that's not exactly a mainstream discussion, but it gathered a lot of people at radiation oncology meeting, primarily radiation oncologists. And before and after the dialogue, I've asked the same question, is how prepared do you feel to integrate radioligand therapy alongside radiation therapy in that same meeting to that same audience, and see that the upper bar is reflecting the answers before the discussion, and the bottom bar actually answers after the discussion. And you see how much it changed with the same people in the same room just by having the dialogue. I know that's not exactly science and very representative, but nevertheless, it shows the importance of the dialogue and something that can be also harvested for the benefit of the topic that I'm speaking about. The next thing is we're living in the digital world, And when we speak about data, I think it's impossible not to talk about harnessing the benefits of digital transformation potential. Radioligand therapy is a complex workflow. There is a therapy selection, planning, delivery. A lot's been said about the quantification and the therapy monitoring already today, so I don't want to repeat this. And then the therapy follow-up. There is multiple stakeholders, there is multiple tasks, handshakes, and a lot of data. But this is good because that's where computers, that's where foundational models, large language models, AI potential can really be harvested. And I think that altogether, looking at how can we better be supported is indeed impossible without integrating those benefits into the workflow of radiotheranostics departments. Coming in the last of my slides, building partnerships. I think no task is complex enough for the capable team, and that's exactly what we're trying to do. So also building partnerships with centers of excellence, whether that's research collaboration or working on the better staging and quantification of the disease, or just working on the education, training, and bringing in the potential to more people to be involved, engaged. And I think that this is also something that stands for the importance of those centers of excellence. So finishing my talk, I would like to say that there is multiple challenges. They're administrative and clinical. There are also patient's response prediction is still a challenge in my view. There is financial availability, like an outcomes-based model agreements potentially coming our way. But they can be resolved with strengthening the role of radioligand therapy through building partnerships, promoting meaningful dialogues, and harnessing digital transformation and AI potential, and altogether, one going into another and back, is that data and evidence-driven individualization, personalization, and predictive models are going to be a help. And with this, I very much thank you for your attention. I've left 1 minute, but I'm happy to take questions at the reception and give this minute to the next speaker.
Our last, certainly not least speaker, sorry. Is Madeline Brown. Madeline Brown is third-year pharmaceutical science PhD candidate from Ray Riley's lab here at U of T. And her work is mainly around radiation nanomedicine, utilizing Auger electrons and radioisotopes. And the main topic she's focusing on is glioblastoma. She has received, if I read that all here, there's more scholarships than I had in my entire career. No, seriously. So she received scholarships from the STAR 21 and Center for Pharmaceutical Oncology, Pfizer Canada Graduate Fellowship in Pharmaceutical Sciences, MDS Nordian Graduate School, and she's been funded by Canadian Cancer Society, NSERC, CHR, and all that together also in collaboration with Triumph and UBC. Very impressive for a third-year PhD candidate. So please welcome Madeline Brown.
Thank you very much. I just want to thank the organizers for allowing me to speak here today. So today I'll be talking about one of our programs that's run out of Dr. Raymond Riley's lab at the University of Toronto. And this is developing a radiation nanomedicine for GBM treatment or glioblastoma multiforme treatment. And the specific project that I'll be focusing on today is how we exploit biological and physical targeting for better treatment outcomes. So just a quick overview into what we will be going over in this presentation. So GBM really presents a unique opportunity for dual targeting of radiopharmaceuticals. This dual targeting approach, if used correctly, can limit off-target effects of the radiation. It can also maximize the radiation dose to the tumor, and it will also improve overall survival outcomes potentially. So why GBM? Why does GBM offer this dual targeting opportunity? So GBM has accessible molecular targets. So 50 to 60% of GBM cells overexpress EGFR as compared to the normal brain tissue. Similarly, EGFR, as we know, is implicated in GBM cell proliferation and tumor growth. And this really makes for our GBM a good candidate for biological targeting of a radiopharmaceutical. On the same line of questioning, GBM is anatomically confined to the brain. And we know that typically recurrence will happen within 2 centimeters from these surgical margins. And this presents a unique opportunity for a local delivery strategy. And this makes GBM a very favorable candidate for physical targeting. So this project specifically utilizes OJ electron therapy. So OJ electrons, as some of you may know, have a very low energy. And they're emitted over very short subcellular ranges, which results in a very high linear energy transfer. And this makes them some of the most potent and precise forms of radiation for cancer treatment. However, one of the limitations of OJ electrons is that given their subcellular range, in order for them to irradiate the cancer cells or cause damage to these cancer cells, they need to be effectively targeted to the nucleus near the DNA and cause these potent DNA double-strand breaks. So here we've developed a mechanism essentially to target them to these GBM cells. So we use this EGFR target that I spoke about before, and we go after it with a radiopharmaceutical that will then attach to the EGFR, and it will be internalized because EGFR has a nuclear localization sequence, and it will be sequestered to the nucleus of the cells. So in this project specifically, we're utilizing mercury-197, and this is an OJ electron that's produced in Triumph, and it's produced by bombarding a gold target with protons, and then this is converted into mercury-197. It's got a very short half-life, but perhaps the most important characteristic about mercury-197 is it can form an amalgam with gold. So we're able to form these mercury-197 gold amalgams. We can then infuse this radiation nanomedicine directly into the brain, and we can use catheters that are placed within the GBM tumor to do so, allowing for a local delivery approach and bypassing the blood-brain barrier entirely, which limits a lot of therapeutics for GBM. And this is a method called convection-enhanced delivery and is a physical strategy of targeting. So we constructed 2 different radiation nanomedicine constructs for GBM in order to test out our theory. So we have a mercury-197 gold nanoparticle, which was unmodified. It has no biological targeting, and it is just administered via convection-enhanced delivery. And this really allows us an opportunity to understand the importance of using physical targeting on its own. Similarly, we constructed a mercury-197 gold nanoparticle that was conjugated with panitumumab, which is an anti-EGFR monoclonal antibody, and administered this via convection-enhanced delivery as well. And this gave us an opportunity to understand the importance of biological and physical targeting when used in combination. Does this improve outcomes? So first, we just went on to test if the biological targeting was effective in vitro. So what you can see here is we have our EGFR targeted mercury-197 gold nanoparticle and the unmodified construct. And we saw that there was much higher activity per cell in human GBM cells when we used our targeted construct. Similarly, on the opposite panel, we see the amount of DNA double-strand breaks. So those are represented by the bright green foci that are seen on the screen, and we see that the EGFR-targeted mercury-197 gold nanoparticles are getting into the cells and causing those DNA double-strand breaks, as compared with the unmodified construct or the control, we aren't seeing the same amount. We went on to test our physical targeting using the convection-enhanced delivery that I spoke about earlier. And so we imaged these using SPECT-CT imaging. So we have here our EGFR-targeted mercury-197 gold nanoparticle and our non-targeted mercury-197 gold nanoparticle. And we saw, irregardless of group, whether we used biological and physical targeting or physical targeting on its own, we saw that the physical trapping of the gold nanoparticles occurred in the brain. We did not see any redistribution to other organs, and if we, up to 7 days post-infusion, and if we zoom in on the head alone SPECT CTs at 7 days post-infusion, we do see some noteworthy differences. In particular, that there is slightly less redistribution of the EGFR-targeted mercury-197 gold nanoparticle as compared with the unmodified construct. Now, this may be, one suggestion for why this might be, is that the EGFR targeted is actually binding to the EGFR receptor on those human GBM cells, and this is causing a less diffuse distribution. However, this needs to be further tested to confirm. So here we're just showing the conformal dose deposition in GBM tumors that we're able to get using these types of radiation nanomedicine. So as you can see in one panel, we're showing that the tumor versus the normal brain, the percent injected dose per gram. And we see that irregardless of group, the EGFR targeted and the unmodified construct both maintain far higher percent injected dose per gram in the brain as compared to the normal brain, over 100-fold greater. When we looked at the dose volume histogram, we saw that the EGFR-targeted mercury-197 gold nanoparticle was able to deliver over 190 gray to greater than 60% of the tumor mass. And with the unmodified mercury-197 gold nanoparticle construct, we saw that only 1% of the tumor mass received this greater than 190 gray dose. However, and perhaps most importantly, is the safety of this type of approach. So as we move 1 millimeter from the tumor margins, we were able to see a very steep dose decline, represented by the dashed lines on the graph. What we can see is the majority of the adjacent brain, 1 millimeter from the margins, is receiving less than classic external beam radiotherapy, which would typically be used for these types of patients. So just to bring it all back, why does this matter? So the impact of dual targeting on treatment outcomes. So we used a human GBM model in mice, and when we treated the mice with the control groups, it's a very aggressive model. They typically only survived, so the median overall survival was only 32 days. However, we did see a modest increase in survival when we used the physical targeting alone, so this unmodified mercury-197 gold nanoparticle. And the increase was out to 43 days. However, the best survival outcomes were seen when we used both biological and physical targeting in combination. So this was our EGFR mercury-197 gold nanoparticle, where we saw a survival increase out to 58.5 days, almost double that of the control group. And this is just further exemplified by the representative 34 days MRI, where we can see that the best tumor control was had by the biological and physical targeted mercury-197 gold nanoparticles. So I'm sure many of you are clinicians in the crowd, and you might be wondering, well, what's the feasibility of translating these types of radiation nanomedicines into the clinic? So we are not the only group who has had this idea. Actually, a group that's run out of San Antonio by Dr. Andrew Brenner has been working on this type of radiation nanomedicine and delivering it via convection-enhanced delivery. So there are a lot of major differences between the study, but they've been working with a beta particle emitter, which is encapsulated in a liposome, and they deliver it via convection-enhanced delivery, which again is this physical targeting that I'm talking about. This is the RESPECT trial, and these are just some of the results that they've seen, but as we can see, their clinical results for their SPECT-CT images 5 days post-infusion look very reminiscent to our preclinical studies. Similarly, their Kaplan-Meier overall survival, these were done in recurrent GBM patients, and if the patient received greater than 100 gray to the tumor, their median overall survival was 17 months, which is pretty impressive. So just a few key takeaways from this talk. So GBM really offers a unique opportunity to exploit both biological and physical targeting strategies. This dual targeting approach that I spoke about was most effective for treating GBM in a mouse model. And this could be a new or novel approach for treating small volume residual or recurrent disease in GBM patients, which we are really excited to continue pursuing. And with that, I would just like to acknowledge a few people, in particular, Dr. Raymond Riley, who's my supervisor and gave me the opportunity to come here and speak today, Dr. Zhongli Kai, who's an expert in dosimetry and helps with all of this research, and to the team over at Triumph, who produces all of the mercury and the isotopes that we use in our lab, and to the STAR team, who does all of our small animal imaging. So thank you, and I welcome any questions if I have time.
I think we have, again, time for one question, if there's one. Otherwise, I have one. Oh, it's there, sorry.
We have. So one of the things that we use is we don't use a chelator because we're forming an amalgam with the gold nanoparticles. But one thing that we've considered using is a longer range beta particle. And we're potentially looking into gold 198 as a new translation. Yeah.
Thank you. I still have one, sorry.
Okay, yeah.
So you said that could be for like a small residual tumor maybe, and there are clinical studies already. Do you have a feeling how far it is away, like for patients here locally, like from your lab, this like dual target approach, like in a clinic for a clinical study?
In terms of our particular study, it's still in the works, so we haven't actually developed. So we do have a GMP facility at our lab at University of Toronto, but we haven't worked towards the clinical development yet. I think likely what will happen is we'll test with a few different radioisotopes, choose our key candidate, and then progress that one to the clinic.
Great. Thank you very much.
Thank you.
And with that, that was the closer of this session. There's one last to go, obviously. And I would like to thank again all the speakers. Great talks, impressive information, and thank you all again for coming.